how does a cell know which genes it has to turn on and when does it have to turn them on? So like we said, cells have all the genes in their DNA are exactly the same, but they're not using the same genes at the same time. So cells need to know which is the pathway that they're about to take, and they do that by responding to signals that they receive from the external environment and from other cells and even from themselves. So cells need to be in constant communication so that they can respond to stimuli from the outside and from other cells in the organism. So the principles of cell signaling first start by the reception of the stimulus. So that stimulus is usually a molecule that acts as a signaling molecule. That signaling molecule binds to a receptor molecule, which is usually a protein. That receptor molecule then relays that information to another set of proteins, and that is called transduction. As multiple proteins will react to the stimulus and pass on that to the next protein. And at the end, you have what is the expected response of the cell. And response usually comes in two ways. One is an immediate response, will be the activation of proteins that are already present in the cell, or the deactivation of proteins that are present in the cell. The proteins can be quickly turned on and off by adding a phosphate group or removing a phosphate group. So this is called phosphorylation or dephosphorylation, and that changes the shape of the protein, so that makes the protein either active or inactive. So that would be a quick way for cells to, if they have proteins that are already made, they can activate them or deactivate them by either phosphorylating them or dephosphorylating them. But there could be other types of cellular responses, like promoting the transport of vesicles that already contain molecules inside of them and tell the cell that it's time to release those molecules to the outside. And another type of response is the activation of gene expression, turning on specific genes in response to that signal. So we're going to look at the two main types of signaling pathways depend on the type of ligand or the type of signaling molecule that you have. And there are two types of signaling molecules. They can either be, just like we saw before, regular molecules, they can either be hydrophilic, which are water-loving molecules, polar molecules, or they can be hydrophobic, which are fat-loving molecules that don't mix so much with water. And the difference between these two is that hydrophilic molecules cannot cross the plasma membrane. They cannot go through the membrane, they cannot enter the cell unless they have some help. While hydrophobic molecules can cross the plasma membrane and enter the cell without any special proteins. So for molecules that are hydrophilic, that cannot cross the plasma membrane, the receptor molecule is usually located on the surface of the cell. So these are called membrane receptors or surface cell receptors, because they will be located on the surface of the membrane on the outside, so that the molecule doesn't need to enter the cell in order to bind the receptor. While for hydrophobic ligands, since they can enter the cell, the receptor is usually located inside of the cell. So the ligand can just get through the plasma membrane and find the receptor inside the cell. So let's take a look at the case where ligands can cross the plasma membrane and enter the cell. So like we said, in this case, the receptor is located inside of the cell, so they're called intracellular receptors. When the ligand enters the cell, it binds to the receptor, and that initiates a signal transduction pathway. Since the receptor is already inside the cell, most likely it's going to go into the nucleus, and the common pathway for these hydrophobic ligands and receptors is that they go into the nucleus and they bind to the DNA and they act as transcription factors, starting the expression of certain genes. So common signaling molecules that follow this pathway that are hydrophobic ligands are steroid hormones. Steroid hormones such as testosterone or estrogen, those are, or cortisol, they are derived from cholesterol, which is a lipid. So all those steroid hormones are lipid-based hormones, so they are hydrophobic, they can cross the membrane, and they can bind to a receptor and activate gene expression. So this is how steroid hormones can have the effects that they have. And you can see those effects tend to be very long term because they are changing the pattern of gene expression, such as when you start producing testosterone in puberty, you start seeing all those changes that are the result of that, like the growth of facial hair and the changing of your voice and the more accumulation of muscle. This is all a change in gene expression. The other type of ligand are hydrophilic ligands. So these are polar molecules that are water-loving but cannot cross the plasma membrane. So in this case, the receptor is located on the surface of the cell, on the surface of the membrane. The ligand will bind to the receptor without having to enter the cell. And the receptor is a transmembrane protein. So it has a part to bind the ligand on the surface of the cell, but it also goes through the membrane and it has a part of the receptor that is inside of the cell. And that part inside of the cell interacts with other proteins inside the cell to result in that signal transduction pathway. So it will activate multiple proteins inside the cell that will lead to the cellular response. The common cellular response of this pathway is by activating proteins that are already made or deactivating proteins that are already made. But it can also lead to gene expression. So if the final protein that is activated in that signal transduction pathway is a transcription factor, then that can go in the nucleus and bind to the DNA and activate the transcription of those genes. But most of the time, they are activating proteins that are already made, so they will have very quick effects, but very short-lived. So for example, if you are running from a predator, a tiger is chasing you, your body will produce adrenaline. Adrenaline will bind to receptors in multiple organs. So you have adrenaline receptors in your pupils that would make you have more focused vision. In your heart, it will make you beat faster so that you have more oxygen flowing through your blood. It will also make you breathe faster so that, again, you're taking in more oxygen. It will make your liver release more glucose into the bloodstream so that you have energy to run. So all these things are very quick and are the result of activating proteins that are already present. It will take too long to make those proteins from scratch. But if you're constantly running from predators, if you live in a place where you're chased by tigers, then you're going to have an activation of gene expression so that you have more of those proteins made so that when it's time to run away from a tiger, you already have those proteins there that are inactive, and all you have to do is when adrenaline comes in, will activate those proteins. So usually those two pathways of increasing gene expression or activating proteins tend to work together so that one will make if you have more gene expression, you have more of the proteins that you need, so that when the short-term stimulus comes, you can respond better to that stimulus. So to summarize, the two main pathways responding to signals depends on the type of ligand, whether the ligand can cross the plasma membrane or not. When the ligand can cross the plasma membrane, it binds to receptors inside the cell, and usually the end result is to activate gene expression. When the ligand cannot cross the plasma membrane, the receptors will be located on the surface. Now, there are multiple types of surface receptors. We're not going to go into that detail of the specific types, but usually those receptors will have a part that goes inside the cell that would activate other proteins inside of the cell, and that will lead to the response, which usually is activating proteins that are already present or deactivating proteins that are already present in the cell. But it can also lead to gene expression, which will be a longer time effect. So if you are activating proteins that are already made, that's a very fast effect. It will happen very quickly. If you're activating gene expression, then you're making proteins from scratch, so it will take longer for the effect to be visible, but it will also be more long-lasting because then those proteins will be made that weren't present before.